I'm really excited to present my work today on a yet unfinished story that suggests that sphingomyelin serves a critical role in the repair of damaged organelles. So an important tool that we use for our research is an engineered version of the equinotoxin that was originally derived from the sea anemone Actinia equina, which binds to sphingomyelin, and upon binding, it oligomerizes and forms cytotoxic pores. Now, the group of Chris Bird from the Yale University has engineered the equinotoxin in a way that it prevents its cytotoxicity and that it also enhances its specificity for sphingomyelin, really making it an ideal sphingomyelin recognizing probe for biological applications. They've used the probe, for instance, to stain recombinantly for exogenous for the sphingomyelin on the exoplasmic leaflet of intact cells, as you can see here in these two microscopy image. Now, the equinotoxin probe can also be expressed inside cells, as I've done myself in the case of the microscopy image here to the very right, where the equinotoxin, tectofluorescent protein, is expressed inside the cytosol. And as you can see, there's a very even distribution of the probe, which shows that there are no stable pools of sphingomyelin on the membrane leaflet facing the cytosol that can be recognized by the equinotoxin probe. And this really illustrates the asymmetric distribution of sphingomyelin across biological membranes, where the bulk of the sphingomyelin is localized to the exoplasmic or luminal leaflets. However, the sphingomyelin asymmetry can be broken. And one way the asymmetry is frequently broken in nature is via infections with pathogenic bacteria. As you can see here, here for equinotoxin expressing macrophages, which are infected with pathogenic mycobacteria that are able to damage and escape the phagosome. And you can see that there's a strong recruitment of the equinotoxin probe to the bacteria-containing phagosome, which was not the case when I used mycobacteria that are incapable of damaging the phagosomal membrane. Now, importantly, this is also in line with the study published earlier this year by the group of Felix Rando in Current Biology, where they used another sphingomyelin recognizing probe and another set of pathogens. Then another way to break the sphingomyelin asymmetry is through the use of lysomotrophic drugs, such as LLOME here, which permalyze acidic compartments, such as lysosomes or late endosomes. And LLOME treatment of HeLa wild-type cells expressing the equinotoxin leads, as you can see, to a massive recruitment of the probe to damage lysosomes. However, there was only minor recruitment in cells lacking intact sphingomyelin biosynthetic pathway due to a double knockout in the sphingomyelin synthase 1 and 2. Here my presentation always referred to as SMS double knockout. This also serves as a good control for the specificity for the equinotoxin probe. Then another elegant way to break the sphingomyelin asymmetry locally at the site of the plasma membrane in a very controlled fashion is via laser damage. Here we can not only control the site of damage, but also the intensity of damage that we cause. So as you can see here, rather severe damage and then pretty small damage here at the bottom. Now, as you have also probably noticed, the equinotoxin probe is recruited almost instantly to the site of damage, which led us to wonder how the recruitment kinetics compare to other molecules and proteins that have previously associated with membrane damage and repair. And one well-studied marker for membrane damage are lectins that sit similar to sphingomyelin at the non-cytosolic leaflet of membranes and are only exposed to the cytosol upon damage. And these lectins are then recognized by galactins, such as galactin-3 here. And what we found was that cytosolic sphingomyelin exposure precedes the recruitment of galactin to damage lysosomes by many minutes. This also serves as a good control for us to say with rather high confidence that it's actually the sphingomyelin leaking out of the damaged organelles and not the equinotoxin probe going inside, because the equinotoxin probe and the galactin-3 reporter are of about similar size. Now, to further understand the kinetics of cytosolic sphingomyelin exposure, we also compared the recruitment of equinotoxin with the recruitment of components of the Escort machinery, like the CHIM4B here in magenta. as it was recently shown that the escort machinery is rapidly recruited to damaged lysosomes, where it actively also contributes to membrane repair. And what we found was that, yes, the escort machinery is rapidly recruited to damaged lysosomes, but cytosolic sphingomyelin exposure still precedes the recruitment of escort. Now, just to sum up the first part of my talk, we found that analogous to the role of phosphatidylserine that is displayed on the surface of damaged cells as an eat me signal, we found that sphingomyelin is exposed to the cytosolic surface, so the other way around, in response to damage inflicted by either pathogens, chemicals, or light. So we're looking at a rather universal process here. And therefore, cytosolic sphingomyelin is an early indicator of organelle damage that is well detectable before no markers of membrane damage and repair. Now, these findings don't tell us so much about the actual significance of cytosolic sphingomyelin, but we speculated that there may be a reason for why cells put so much effort in maintaining the sphingomyelin asymmetry, and proposed a working hypothesis stating that sphingomyelin may actively contribute to the repair of damaged membranes. To test this hypothesis, I compared the ability of either wild-type or SMS double knockout cells to repair damaged lysosomes. And as a readout system, I used the lysotracker reagent to visualize intact lysosomes and then permalyze the lysosomes with the lysomotropic drug GPN, which then led to a disappearance of the lysotracker signal. However, the signal could be restored by washing out the GPN, as we can nicely see here for the wild-type cells. But what we found was that sphingomyelin deficient cells show a defect in the recovery of the lysotracker signal. Now, the ability to repair damaged lysosomes could be restored in SMS double knockout cells by reintroducing the sphingomyelin synthase 1 under the control of a doxycycline-inducible promoter. The defect in lysosomal repair was also confirmed via a drug sensitivity assay, where we found that SMS double knockout cells are more sensitive to lysomotropic drugs. Okay, so as I mentioned a few minutes ago, studies have showed that the escort machinery is important for the repair of lysosomal membranes. As you can see here in this figure from a paper published in 2018 in Science, where a double knockdown of the escort of two escort components affects lysosomal repair. And we then wondered if our observed sphingomyelin-dependent membrane repair is connected to the escort machinery or whether we look at the escort-independent pathway. And to test this, I performed the same double knockdown of escort components as used in the Science paper, either in my wild-type cells or in my SMS double knockout cells, and tested the effect on sensitivity to lysomotropic drugs. And as you can see here in blue, an escort knockdown in wild-type cells leads only to a minor enhancement in sensitivity, which is not significant. However, the same knockdown in SMS double knockout cells, as seen here in red, gave a strong additive effect in drug sensitivity. So while either knocking down escort or knocking down sphingomyelin synthases by itself has rather a minor effect on repair, removing both pathways causes a massive increase in drug sensitivity, which what we think points towards the direction of two independent pathways. The idea of two independent pathways was also further supported by an observation that showed that SMS double knockout cells show no defect in the recruitment of the escort component SHIM4B upon lysosomal damage when compared to wild-type cells. The only significant difference I found was that SMS double knockout cells show a prolonged retention of SHIM4B on damaged lysosomes, as can be seen here, which potentially could be explained by saying that those cells that lack the sphingomyelin-dependent pathway of repair rely more on the escort-dependent pathway, and the escort needs to take over. Now, to conclude the second part of my talk, we learned that sphingomyelin-deficient cells are defective in the repair of damaged lysosomes, and our results suggest that the sphingomyelin-dependent pathway of repair is likely independent of escort. And therefore, we proposed two different models. Model A stating that sphingomyelin acts as a cofactor for membrane repair machinery, and Model B saying that sphingomyelin acts as a substrate in a membrane remodeling pathway. Now, I've already begun to test the model B. For this, I engineered a tool that targets a sphingomyelinase to the lysosomes by attaching a bacterial sphingomyelinase to LAMP. And the idea was that upon induction of membrane damage, the LAMP1 bacterial sphingomyelinase converts the sphingomyelin that leaks out to ceramide. And this is exactly what we see when we express the construct together with the equinotoxin. We only see the equinotoxin puncture briefly lighting up, unlike what we see when we use the dead version of the bacterial sphingomyelinase. This also, by the way, shows nicely that the equinotoxin binding to sphingomyelin is reversible. I have also already used the construct to test for lysosomal repair and found that, if anything, sphingomyelin turnover promotes repair, but the effect is very minor and not significant, which potentially could be explained by saying that cells have already a well-equipped repair machinery, and now our plan is to disrupt the endogenous repair pathways to increase a potential dependence on the sphingomyelin-dependent repair. Okay, so with this, I'm at the end of my presentation, and I want to thank everyone in my lab and in the Barish lab, but especially yours for being a great supervisor. And again, I also want to thank the group of Chris Bird for support and providing us with the equinotoxin, as well as Yusuf Hanun and Chris Clark from the Stony Brook University for frequently helping us out. Okay, thank you for your attention, and I'm looking for questions or any helpful feedback.